autoimmune diseases are associated with certain HLA antigens. The exact mechanism by which the bodily T cells target the self-antigens that are presented on MHC molecules, however, is yet to be agreed upon. A well-supported observation is that autoimmune diseases often become symptomatic after illnesses, such as common cold or bacterial infections. What is a reasonable explanation for this observation? Well, pathogenic antigens may produce peptides that are closely similar to the self-antigens, with only one or just a few amino acid differences. This is the so-called molecular mimicry. This may induce a strong reactive T lymphocytic response in the host. Another explanation is that the self-antigens that are released from damaged tissues are picked up by macrophages or dendritic cells, and they set off an immune response against self-antigens. This phenomenon or explanation is known as bystander activation phenomenon. Infectious antigens may cause also super antigen-induced activation of certain T cells that are reactive against the self-antigens. Let us summarize and look very quickly over major autoimmune diseases and their HLA associations. As you can tell, with the progresses in the science of immunology, every day that passes by, we add several more autoimmune diseases into the list of known autoimmune diseases and their associated HLA alleles. Now, on the left column, I'm showing the major HLA alleles, and on the right, the associated autoimmune diseases. For instance, HLA-A3, hemochromatosis, B8, Graves' disease, B27 with inflammatory bowel disease, psoriasis, ankylosing spondylitis, and Reiter's syndrome. What is the other name for Reiter's syndrome? Reactive arthritis. The other condition associated is acute anterior uveitis. HLA-B47, 21-hydroxylase deficiency. HLA-DR2, systemic lupus, multiple sclerosis, hay fever, good pastures. HLA-DR3, Graves' disease, autoimmune hepatitis, Sjogren's syndrome, diabetes mellitus. HLA-D4, rheumatoid arthritis, diabetes mellitus, and pemphigus. HLA-DR5, pernicious anemia and Hashimoto. HLA-DR7, steroid responsive nephrotic syndrome. HLA-DQ, celiac disease. And again, these are just some major and well-known associations. There are a lot more associations that we have found, and as I said, every day that passes by, we identify more and more associations. In an immunology lab, the goal is to identify CD3 positive cells from samples of human peripheral blood using polystyrene beads that become magnetic only in a magnetic field. The beads are coupled with anti-human monoclonal CD3 antibodies that are specific for CD3 membrane proteins or membrane antigens. The cells that are bound to the antibodies are then captured and identified. Which of the following cell types are isolated with this procedure? To make it simple, monoclonal anti-CD3 antibodies identify CD3 cells. The question that we are asking is that what cells of the body have CD3 proteins on their surfaces? Is it natural killers? B lymphocytes, T lymphocytes, CD8 positive T lymphocytes, or CD4 T lymphocytes? Actually, the answer is all T lymphocytes. All T lymphocytes, CD4, CD8, and CD4, CD8 cells that are precursor of CD8 or CD4 cells. So in a sense, had you picked up D or E, you would have been partially correct as well. But the most precise answer would have been option C. So all of the T cells express CD3. B cells, on the other hand, express immunoglobulins and CD5 molecules on their surface. Natural killer cells express neither CD3 nor immunoglobulins. However, they express CD16. So let's summarize. CD16 for natural killers, CD5 for B cells, CD3 for T cells. Note that CD4, CD8 cells are progenitor T cells that over time evolve into CD4 or CD8 cells. So all T lymphocytes, CD4, CD8, CD4, CD8 cells express CD3 molecules. Mnemonic, T for tree, T cell for 3. In this cartoon diagram, on this side I'm showing a nucleated cell, and of course you see a one-legged MHC molecule 1 on its surface. Of course it presents an internal antigen, it may be a virus, and pass that or present it to T cell receptor, TCR. I'm showing here a cytotoxic CD8 T cell. Remember, 8, 1. 8 multiplied by 1 gives you 8. And here I'm showing the CD8 molecule. I represent it as number 8. Of course, these CD8 T cells, on their surface, they have many of these CD8 proteins. Also, this is a T cell. It has a CD3 on its surface. It has many CD3s. Actually, to be more exact and to give you a heads up, the CD3s are closely associated, as we're going to see shortly, with TCR receptors. On the right side, I'm showing a dendritic cell. This is an antigen processing cell, and it has a two-legged MHC2 on its surface, and it presents particles of exogenous antigens. Of course, it passes that through T cell receptors, but this time that T cell, that T helper cell, is a CD4 positive, because 4 multiplied by 2 gives you 8. And again, since this is a T cell, it has CD3 protein on its surface. And it has many of these CD3 molecules, but remember one important fact, that CD3s are closely associated with TCRs, not only in CD4 positive T cells, but also in CD8 positive T cells. Let's look a little bit more closely to the structure of T cell receptors. This T cell receptor complex is a little bit more complex, and of course is associated with CD3s. Actually, another mnemonic, CD3 complex has 3 dimeric chains. So CD3 has 3 parts, 1, 2, and 1 intracellular one as well. And they're all associated with T cell receptors that I'm showing this in dark brown color. That's your T cell receptor. And it has an alpha and a beta chain. The beta chain is the one that has the VDJ segments. The alpha chain only has VJ segment. Anyhow, let's go through it together. The T cell receptor is a complex composed of an alpha and a beta chain in conjunction with 3 CD3 molecules or co-receptors. These are the 3 CD3 parts. 1, 2, 3. The three pairs of CD3 dimers are composed of four distinct polypeptide chains: epsilon, gamma, and delta. Epsilon, delta, and gamma. The intracellular tail of the CD3, this is the one. The intracellular tail of CD3 molecule contains a single conserved motif known as immunoreceptor tyrosine-based activation motif, or ITAM. Here I'm showing the ITAMs in blue. ITAMs in blue. The ITAMs are essential for signaling capacity of the T cell receptors. I want you to note that TCR by itself cannot transmit a signal into nucleus. CD3 molecules play an important role in mediating TCR signals into the cells, and in particular, the role of the intracellular CD3 is prominent with this respect, because this is the one that extends well into the core of the cells. Of course, the other two are also important, but it seems that the one which is more important is the one that penetrates deeper into the core of the cells. What was the first monoclonal antibody that was approved as a drug in humans, and what does it do? This is muromonab, or orthoclone, or OKT3. Muromonab. What does orthoclone do? It targets or binds CD3 receptors that are required for T cell activation. It is mainly used for therapy of acute glucocorticoid resistant rejection of allogeneic renal, heart, and liver transplants. And we're going to be covering it from clinical angle a little later. Other anti-CD3 antibodies that are developed are otelizumab, which is also known as TRX4, teplizumab, MGA 031, vezilizumab, which is also known as Nuvion. Note that all of these are monoclonal antibodies, that is, they are clones of a single parent cell. And what they do all is that they can bind CD3 molecules on the surface of T cells. These monoclonal antibodies, they have shown promise for treatment of Crohn's disease, ulcerative colitis, and type 1 diabetes. In this table, I'm showing the major therapeutic monoclonal antibodies. To just orient you, the suffix MAB denotes the whole monoclonal antibody. Note that monoclonal antibodies can also be made against the Fab or the conjoined Fab fragments. EpCAM stands for epithelial cell adhesion molecule. It is shown that it is oncogenic and upregulates c-MEC. CD52 is a protein on mature lymphocytes. CD152 is protein associated with cytotoxic T lymphocytes. CEA, it stands for carcinoembryonic antigen. So these are the abbreviations that you're going to see for the target proteins. So let's go very quickly through this. At least I want to help you to pronounce these terms a little better. Abciximab is the monoclonal antibody against Fab. The target of this monoclonal antibody is CD41 or integrin 2B, and the usage is for inhibition of platelet aggregation. so it has antiplatelet function. So it targets the platelets. The other one is adalimumab, which is again a monoclonal antibody and targets tumor necrosis factor alpha. The usage is for rheumatoid arthritis, Crohn's disease, psoriasis, and management of ankylosing spondylitis. Next one is aducanumab. Target is EpCAM. Usage is for prostate and breast cancer. The next one is alemtuzumab. Target CD52. Usage chronic lymphocytic leukemia and multiple sclerosis. Blind A2-mumab. Target is CD19. Usage second line treatment for ALL. Ipilimumab. Target is CD152. Usage management of melanoma. Lebrikizumab. Oh, these names are so difficult to pronounce. I don't blame you if you can't. This one targets CEA for management of colorectal cancer. Lebrikizumab targets surface antigen in hepatitis B. Of course, we use it for the management of acute or chronic Hep B. Obeltoximab. Target is Bacillus anthrax. Usage for patients who are exposed to anthrax. Zatoximab. Target is HER1 protein and usage is for breast cancer that are related to HER1 gene. Cancer cells often express reduced MHC1 receptors, and as a result, they tend to avoid destruction by cytotoxic CD8 T cells. Which of the following immune cells is most effective in causing apoptosis on cancer cells that lack MHC1 molecules? Neutrophils, macrophages, dendritic cells, natural killers, or T helper 2 cells. The correct answer is natural killers. Just to know, NK cells express 2 sets of surface receptors to identify virally infected cells or cancer cells. One of the 2 sets kills the affected cells and the other inhibits the killing. Killer inhibitory receptors or killer cell immunoglobulin-like receptors or KIRs are expressed on NK cells. These receptors are members of immunoglobulin gene superfamily that are specific for MHC1 molecules. KIR receptors can distinguish between major histocompatibility complex class 1 allelic variants, which allows them to detect virally infected cells or transformed host cells. Most KIRs are inhibitory, meaning that their recognition of MHC molecules suppresses the cytotoxic activity of their NK cells. Underproduction of MHC1 molecules, which is characteristic of most cancer cells, or their structural modifications, which is characteristics of cells that are virally infected, helps these cells to avoid destruction by CD8 T cells. So CD8 T cells can only identify proper number and proper quality of MHC1 molecules on different cells. If the cells modify the MHC number or MHC qualities, the CD8 T cells are unable to destroy those cells. However, at the same time, the MHC changes removes the inhibitory effects of natural killer cells, and they become susceptible to natural killer cells. So here is the important thing: the infected or virally infected cells at times may avoid the CD8 positive T cells. However, they cannot avoid natural killer cells. The natural killer cells also express CD94 NKG2 receptor. This receptor will allow them to identify modified MHC1 cells on tumor cells and virally infected cells. Let's talk a little about natural killers. Natural killers play a vital role against virally infected cells and cancer cells. They react within 3 days against virally infected cells. The only phagocytic cells of the innate system that manifest qualities that are characteristic of the adaptive system is the natural killer cell. They are evolved from lymphoid progenitor cells that make B and T cells. However, unlike T cells, their receptors are germ-like encoded. This one makes them similar to neutrophils or macrophages. Natural killers account for less than 2% of blood lymphocytes. Uniqueness of these cells is that they can identify stress cells in the absence of MHC. I want you to note that virally infected cells that do not express MHC may avoid the adaptive cells or the cells of the adaptive system. However, they cannot escape the natural killer cells. NK cells possess intricate mechanism to differentiate self from non-self. They are trained to leave the self intact and attack the non-self molecules. Their ability to discern between stressed cells and normal cells is controlled by a balance of inhibitory and activating signals within natural killer cells. Activity of NK cells is enhanced by interleukin 12, interferon alpha, and interferon beta. Don't confuse natural killer cells or NK cells with NKT cells, also known as natural killer T cells. These terms are only there to confuse medical students. These two cells, NK cells and NKT cells, are phenotypically and from a standpoint of origin different from each other. NKT cells often activate NK cells by making interferon gamma. In contrast to NKT cells, the NK cells do not express TCR receptors, which we earlier said is the famous pan-T marker. Means that you see it on every T cell. They don't also express surface immunoglobulins or B cell receptors. What do they express? They express CD16 and CD56 instead. Let's look more closely at natural killers' inhibitory and stimulatory receptors. I'm going to be referring to these two cartoon diagrams for you. Natural killer cells constantly survey the blood, lymphatics, and tissues of the body in search of transformed or pathogen-infected cells. On healthy cells, this diagram, the inhibitory receptors, I'm showing inhibitory receptor here, the inhibitory receptors or KER receptors recognize self-MHC1 complex sort of molecules, and this will activate an intracellular pathway within the natural killer cells, and it stops cytolytic NK cells response. So identifying the self-antigens inhibits the response of the natural killer cells. At the same time, activatory receptors are designed to identify unhealthy proteins on the surface of MHC1 cells. On the cells that are normal, these receptors at the same time, they do not feel any unusual proteins such as MICA stress protein that is expressed on the cancer cells. Therefore, they will not initiate an attack response. So there is a balance and check on the natural killer cells. We have a combination of inhibitory and activator receptors, and the two balance each other so that things don't go out of control and the natural killer cells do not become vicious to attack healthy tissues of the body. On the stress cells that I'm showing on the right side, or infected cells, inhibitory receptors, they sense something fishy, and they sense absence of constitutively expressed self molecules on MHC1 cells. This time, this reverses the inhibitory receptor's function and leads to a killing response. Meanwhile, the activator receptors on these cells also sense abnormal surface molecules. This may be presence of the MICA stress protein in the cancer cells. This leads to activation. Net result is release of granzymes and perforins on the target cells and the apoptosis of the target cells. Note that some authors believe that the response of natural killers in killing or letting go of the target cells is the net result of positive stimulatory and negative inhibitory signals that the nucleus of these cells receives from the surface receptors on the natural killer cells. Note that KIRs, or killer cell immunoglobulin-like receptors, is one of the descriptive terms that is assigned to these natural killer receptors. KIR inhibitory receptors signal through immunoreceptor tyrosine-based inhibitory motif in their cytoplasmic domain. Activator receptors do not have items and use other type of KIR receptors. To summarize, natural killers with their KIR receptors and CD8 positive T cells with their TCR receptors bind to MHC1 molecule. However, this interaction activates the CD8 cells, whereas it deactivates the natural killer cells. We talked about MICA. MICA stands for MHC class 1 polypeptide-related sequence A, if you have been interested where the name MICA comes from. But in my opinion, cartoon-wise or caricature-wise, MICA stands for microphone. It's a microphone inside the stress cell who shouts from inside the cell and broadcasts the noise that help me, I'm stressed out. That will help you to remember it as a mnemonic. So MICA for my CA for my cancer. So you see MICA being expressed in the cancer cells. That gives you a very good mnemonic to remember this concept. Anyhow, MICA is a cell surface glycoprotein associated with alpha chain of the MHC1. And as you see in this diagram, the microphone is next to the alpha 1. Remember, the alpha 1 has a transmembrane leg. So this is associated with the alpha leg of the MHC1. And it includes a transmembrane segment and a cytoplasmic tail. And of course, this is the cytoplasmic tail of it in a cartoon fashion. MICA acts as a stress-induced ligand for NKG2D receptors of natural killer cells that have the ability to broadly recognize MICA. Engagement of the NK cells induces cytolytic responses. It is postulated that tumor cells and stress cells induce MICA, and MICA attracts natural killer cells. This is, in my opinion, the most important thing that you need to remember for your examination. NKG2D is a major recognition receptor for detection and elimination of transformed or cancerous cells or infected cells. In NK cells, NKG2D serves as an activating receptor and triggers cytotoxicity. Note that CD8 positive cells also express NKG2D, which sends co-stimulatory signals to the nucleus of those cells.